Okay, so 59 seconds precisely. Thank you for coming back. If you did, I hope you have coffee. I wish I did. I was actually editing a couple of slides. Let's continue. So, as I mentioned earlier, I wanted to create a clear differentiation between computational neuroscience and neural computation. These terms are used interchangeably often in this field. And I feel that to keep the mind clear about what are the goals in the minds of each scientist, it helps to try to attribute to them what is the direction that they're taking in this immense problem space, as we've seen a second ago. So in this mind map here, I put them diametrically opposed, and I wanted to elaborate a little bit more about what their goals are in between this neural computing field, more machine learning related, and this computational neuroscience field, more simulation related. These are not sharp boundaries by no means, and some people might not even completely agree with me on these definitions. And yet, I decide that it's important that we align our heads to what are the important dimensions of these problems. And so this is why this slide exists. It's a disambiguation between computational neuroscience and neural computation. All of them is looking into the brain. All of them will say, we're trying to explain the brain. All of them will say, it's important to understand how neurons work, although to different degrees. The computational neuroscience scientist will think that the level of modeling he needs is higher, so the resolution is higher. The abstraction level is it's at a low level, at the level of the implementation, the ion channels, the action potential. Whereas in neural computation, we're going for the cognitive functions, like the pattern recognition, the decision making, the language. So the focus is slightly different, even though they cross talk. You will notice that the dashed line I put in the middle is very faint. To give a sense that these are dimensions, right, between computational neuroscience and neural computation, there's some sort of continuity there. And if you take different scientists in the field, they might be at different points for each of these dimensions. But let's discuss them one by one. So the focus on computational neuroscience is experiment. You look into the cell, you have a patch clamp, you look at the actual properties of the neuron. Whereas for neural computation, you're engineering from neurons. You're measuring a lot in computational neuroscience. You're measuring the physical underpinnings of the neuron. You're measuring capacitance, conductance. Whereas in neural computation, you're more like, okay, so what do we need to know? So what do we need to assume in order to be able to produce a function? We're trying to explain something. So if we want to explain pattern recognition, what do we need? What are the bare essentials? Some of your answers on what's the right level of modeling suggest that you are thinking along the lines of the more simplified, the better, because you get more explanation power out of it. That's the level of neural computation. However, you're not so concerned in making hypotheses about how actual cells work. So how about you change the temperature of maybe climate change? And how do brains change? So reptiles, like under the sun, they like to languish. And whereas mammals stay at 37 degrees. What happens if you put a mammalian cell at 40 degrees, close to a fever? I mean, you change the dynamics of the brain. That is hypothesis testing. It relates to changing a certain parameter and seeing what's happening at the other end, at the level of biology. So computational neuroscientists focus on biophysics. Neural computation focuses on biobehavioral function. Computational neuroscience, this is an important one. Plasticity and learning. Those are terms that are often used mixedly in a hodgepodge. But plasticity is effectively the physical change of something, whereas learning is what we acquire, right? So we've learned to juggle. You've learned to speak Mandarin, but your neurons are undergoing plasticity even if you don't learn anything. If you're in the couch, thinking about nothing, your neurons are changing. There is plasticity. Plasticity becomes learning if it's in a particular structure. So in neural computation, the focus is in how do you change the neurons in a particular way so that function is acquired. Whereas in computational neuroscience, is how do neurons change, right? At the level of the synapse. If I have activity in the presynaptic neuron and in the postsynaptic neuron, how do they change? So this is the interface. Computational neuroscience looks into the anatomy, thinks about basal ganglia, cerebellum, cortex, hippocampus. Neural computation thinks about circuit, input, output, things, first layer convolution, second layer decision making, third layer recurrent neural network. So how do you put these things together? Assembly. So that's why then there's these language differences, although people can exist in both again. So language is physics and neurodynamics for computational neuroscience, whereas computation, like in computer science, for neural computation. A lot of people working on neural computation come from a computational neuroscience background. Computer science backend. And the computer science backend focuses on abstractions and objects, like in Python. So they're using their metaphors to try to understand what the brain is. They might even understand, quite a bit about psychology, and still what the language that they use is the language of computation. So Turing machines, right? Logics, propositional logic. The details in computational neuroscience matter a lot. So these implementational details, whereas in neural computation, they matter less. Dimensions are many or few. The computational cost, this is for creating a simulation, right? If you think about the comparatively, the computational neurosciences, if we want to simulate the brain at the level of computational neuroscience, it's way, way more math, machinery, more computation underlying the simulation than neural computation. If you have 1 billion neurons in neural computation, these are simple neurons, and it's going to be relatively straightforward in a powerful computer to simulate them. Whereas if you're wanting to really simulate the brain, you probably need a cluster. So questions in computational neuroscience are metrical questions, measurement questions, how much, how, so hypothesis testing, if I do this, what happens there? Whereas in neural computations, why does, when's better? So where from does this function emerge? What are the bare essentials that I need to assume? And so the bottom line, simple. One's simulating, the other one's computing. Bear in mind that I, for example, am a computational neuroscientist, but I am interested in neural computation. I think it's interesting to try to understand what are the details in biology that can translate into novel ideas of neural computation. So historically, the main ideas underlying neural computation from biology, about 4 or 5, and we're going to be talking about them across the course. But the exchange moments were actually relatively few. So there's not a lot of biology that has been taken into neural computation. There's a little biology that has been taken into neural computation, and is immensely powerful. I think that, as many of you have pointed out, that the interesting bit is the intersection between the two, where one informs the other, where we learn how the brain works and then can apply into computers. So right in the middle. The course here presumes to try to give you a sense of that. So this is a model in computational neuroscience. It's a Purkinje cell, and it has cylinders representing the pieces of dendrites and a sphere representing the soma, and it computes activity, electrical activity across the membrane. This is a neural computational model. It integrates inputs that are real numbers. It sums them. It produces a nonlinearity function. So the activity AI equals to a certain nonlinearity of the total input, and that's the output, right? So this creates the dimension. Neural computation is this guy. Computational neuroscience is the highly detailed compartmental geometrical model. And how it all began, let me talk a little bit about computational neuroscience first, and then I'm going to give you some other examples of neural computation, particularly one implementation of called Mango, of a cognitive model. But this paper, which was not, by the way, in 1990, this is a reprint from 52, is the paper that gave Hodgkin and Huxley their Nobel Prize, and is the paper that tried to capture the biology of the action potential in a mathematical physical model for simulation. So it's really the dawn of computational neuroscience in 1952. It's the first attempt to really simulate first successful attempt of simulating simulating a neuron in its biological implementation. It talks about ions, sodium, potassium, chloride, calcium in other cases. And it has come a long way. So from 52 now to 2020. In 2020, actually this paper is from I think it's 2009. Can you see it here? Or 2012? 2015. So look at the number of authors. It's just like cosmological levels here. This is a reconstruction of an entire piece of mouse cortex with as much detail as possible. Some of you may have heard about the Human Brain Project. So this is the onset of the Blue Brain Project. It was before that. But in this paper, 2015, they needed all these people to elaborate on the Hodgkin-Huxley compartmental model to recreate individual cells across the whole brain. For example, here you have a vast variety of different neurons, both excitatory and inhibitory neurons with their dendritic and axonic trees that had been reconstructed from actual anatomy to put them all together and see what they act like. So here you have in panel A, you have basically a plot of the activity of the single dendrites, where the color is the membrane potential of the single dendrites along a column. The scale here, where you see the Roman numerals 1, 2, 3, 4, 5, and 6, these are the layers of the cortex of the mouse. So it's about 800 micrometers thickness. Is that in vraag voor mij? Oh, no, I guess not. So then in B, you see what we call a raster plot. On the x-axis, you have time, and each single marking is one action potential from a cell at that level of these of the cells in A, B, and C. And so you see in C, panel C, all of the different populations of cells indicated by their code names. So for example, here L23 NBC. So that's a layer 2-3 basket cell. No, so or you have a L5 TTPC. So layer 5 thickly tufted pyramidal cell. And so you see that they have different activities, different types of action potentials are constructed. So all of this based on the Hodgkin-Huxley model we just saw. So every single cell here in this model is like an Hodgkin-Huxley cell, actually every little piece of dendrite. And from this model, we, let me actually show you the progression of the activity. So this is a 5-minute video. So this is a reconstruction of the activity for the figure I just showed you. Neurons appear or neural models appear at the rate that they become active. So here you have a single neuron active first, and that neuron is sending synapses to other neurons, activating some dendrites, and suddenly some more neurons come in. So that neuron had been stimulated. So the network then starts picking up activity, and more pyramidal neurons show up. And the colors represent membrane potential, so the hotter colors represent the activity, and suddenly there are bursts of activity, and you see a lot of neurons firing at the same time. Might definitely argue about certain details on this kind of super detailed compartmental modeling. And so there's a lot of the important details that I'm leaving aside. But I wanted to give you a sense of what the path of where computational neuroscience started from Hodgkin-Huxley beginnings all the way to reconstructions of entire cells. Here, this is a model of a slice representing an experiment, and this is a beautiful prediction, absolutely stunning prediction. I'm going to get to. So it actually talks about a phenomenon that had been observed but remained unexplained in the slice. I guess that you haven't had enough neurophysiology to really, but you can get the sense. So the sense is this: when the neurons become active, they become active as a group, and they all oscillate together in the slice. So we can explain that via the recurrent feedbacks in the Hodgkin-Huxley neurons. Probably can create a simpler example, but the fact that this complex example is able to reproduce that, assuming only the structure of the neurons, is pretty impressive by itself. So you see some neuronal propagation, some waves. The video is long. I'll provide the PPT with a video in Brightspace. You can look at it more calmly, and there's actually also an audio track. And going back to what computational neuroscience is aiming at, it's predictions. So these are the predictions from this particular paper. So the market circuit predictions. These are novel predictions that have not been done before on the basis of having reconstructed this neural network to a very detailed level, trying to reproduce single neuron activities. I'm not going to go into them. It's just fun that you can predict or produce 18 predictions out of this thing. At least, you know, you put all these people to work together, and you've made this massive model, like cosmological scale, and there are effective scientific predictions with all of the unknown parameters and all of the difficulties, empirical difficulties of reconstructing the circuit. You can still get to the bottom of things. But that's not it. You can actually go the other direction. Say, okay, but give me the essentials. So let us try to, how much detail can we ditch, and what can we still keep? So this slide represents that thread. And so in level 1, you have highly compartmentally detailed models with ion channel distributions. In level 3, you have single neuron models. Maybe here in this case as a Hodgkin-Huxley, single compartmental. Could also be an integrating fire model as a single compartmental model. A cascade model is something that is more often used, or level 5, like box model in neural computation. These are models that try to emulate the activities of entire populations of neurons. So going into this level of abstraction, we find ourselves again in the realm of neural computation. We pick up smaller, simpler components, and we try to see whether we can explain cognitive function, such as pattern recognition, categorization, knowing that this is not that, object tracking, you have like a ball in your visual field and you want your eyes to follow it, motion perception, you want to know that you're in the train, we know that see the thing moving, that is computation you're undergoing. That's question. How do you do that with neurons? How do you know that it's the train moving and not the world? So these are problems of neural computation, trying to reproduce that kind of cognitive function and essentials via simplified neurons. Using the model we just had a moment ago from the Human Brain Project, in the question of how does motion perception work, is in all likelihood overkill, because there are way too many details. Will be hard to put it together. Maybe in the end we would like to have super complex models that also do motion perception to see what else they can do in comparison with the simplified models. But if you take the simplified models and you ask the question, how does motion perception work? This is how we would go about it. You have then here Also motor control, creating patterns of motor control, navigation, ability of going through a shopping center or shopping mall and then finding the shop again, producing language. So focus on these ones. And then the bestiary. So here is a guide of modern neural networks where we have different kinds of simplified neural networks, as we were talking about a moment ago. Now, this is in neural computation. This is networks that do stuff according to AI style, machine learning style, and they take into account certain assumptions and some certain simplified views of individual neurons, and many different circuits that have a particular goals in. Someone actually started to blow leaves away, so just a little, okay. Sorry for that. And so what I was guessing at is that someone started producing noise outside. I find it a little bit break the flow of concentration. Apologies. Probably I broke yours now. So you have the long short-term memory networks, for instance, that produce language sequences. You have the feed-forward neural networks that produce pattern recognition. You have convolutionals and deep convolutional neural networks. And so these, from neural computation, trying to capture those structures of function relationships, as I mentioned earlier. All of this comes from that simplified neuron I showed you a second ago. So here's pattern recognition. You have an example of a perceptron. And so a single neuron, which is represented here by function omega, is the onset of the idea behind deep learning neural networks. So in deep learning neural networks and in feed-forward neural networks, in neural computation, you see a lot of these kinds of processing schemes that relate to the hierarchy of processing in our brains. So here you have V1 is a visual brain area that has the ability or has cells that respond to orientations of bars in the visual field. In V2, you look at the edges. In V4, you look into outlines and combinations like objects, whereas in inferior temporal posterior cortex, you will have morphological knowledge of words, so basically semantics, really. Whereas in IT anterior, you have more conceptual abstractions. So artificial intelligence and neural computation are trying to inspire themselves from findings of the brain where we seem to observe certain hierarchies and translate those hierarchies into operational networks. To go back, the difference between. So this is a slide about transduction, the idea that in neural computation, you're taking very closely into account the fact that patterns are coming in, right? And they're converted from physics through the sensory systems into percept. So from photons in the eye, you get vision, complex object pattern recognition, and so forth and so on. I won't belabor this point. I'll come back to it in lecture 5. And to close this, I wanted to give word to a prominent researcher in the neural computational field, Chris Elias-Smith, who has made a model. And I'm going to introduce you now to his model by playing this little video. Can you hear the video? Let me know if you can hear, because no, okay. So like I think that I have to actually to share the in Zoom. I probably have to share the entire same system. Ooh. So. There's a little problem to let me try one thing. If it's too bad, then you let me know. I'm going to try to select the microphone from the computer and having the computer listening to what the computer is saying. Else I will have to remap the
My colleagues and I at the University of Waterloo have built a large-scale model of a functioning brain called SPAWN. The model receives its only input as a 28 by 28 pixel image and controls a simulated arm. In this video, we see both the activity of the various brain areas and decodings of the neural representations of those areas. The first task is a recognition task. SPAWN writes the digit it sees in the image. Individual neuron voltage spikes are shown scrolling through the thought bubble at the back of the brain. The decoded value is displayed on top of that activity. The second task is called copy drawing. Instead of just recognizing the digit, the model must reproduce the style of the digit. Here it successfully copies two styles of the number 2. SPAWN's representations are thus useful for both categorization and encoding fine perceptual details. The next task is a counting task. This task demonstrates that SPAWN can not only represent and categorize numbers, it also has an understanding of numerical concepts like order. SPAWN is first shown a number to start counting from, 4, and then it is shown how much to count by, 3. We can see the internal progression of numbers as it counts and finally writes its answer. Next, we have a question answering task. Here, SPAWN memorizes a list of numbers and then answers a question about the list. You can watch the topmost working memory as it stores the items in the order they are shown. In this case, SPAWN is asked what is in position 5. SPAWN answers the question by decoding its working memory and using the result to drive the arm. The final task is a serial working memory task, in which SPAWN must memorize a list of items in order and then repeat it back. We have seen several tasks that SPAWN performs correctly, but the errors it makes are equally important to determining if it is a good model of human cognition. Note that the darkness of the letter in the thought bubble indicates how well that item can be decoded from the current representation. SPAWN is now drawing its memory of the list, but when it gets to the 8, it is no longer confident in its own decoding of that memory, so it draws a horizontal line, indicating it has no answer. If we look at the pattern of errors over many versions of SPAWN, we will notice that it matches the details of human error patterns. We have now seen half of the tasks that SPAWN.
Okay, so
because some of you cannot hear what's coming out of the video, I'll summarize it now, but the video is also going to be put into the Brightspace PowerPoint, and then you can, it's going to be a hefty PowerPoint because it will include 2 hefty videos, about 400 megs. I hope you have that space in your video, but then you can look into the detail of what they're saying. I'm going to, however, explain to you what this is about. So here's a book. It's called How to Build a Brain. It's by Chris Elias-Smith. He had another book before this one called Neuroengineering. He's a researcher in Canada that has put together one of the largest neural computational type models of the brain right now. And what he has the inner persuasion that a good model of the brain must have function. And so he's tried to reproduce by engineering certain beliefs that we have about neurons and brains with integrate and fire networks, certain paradigms on learning. For example, remembering a list of neurons. Remembering a list of numbers. So if you're given like 3, 4, 7, 2, 1, what was the first number in the list? You can write a model that does this via spiking neurons. Not too bad. If I continue the list very big, I myself already forgot the number. So someone actually answered this 3. Yeah. So that you acted just like SPAWN there. This model. And what Elias-Smith is trying to do is trying to embody multiple functions of the brain at the same time, so that they constrain each other, and so that this brain is not simply an AI tool that can tell cats from dogs or tell what is funny, but it actually is able to have a more holistic understanding of multiple tasks at the same time and see the reciprocal constraints of the different levels of the network. It's highly simplified, but it can do many tasks at the same time. It can remember certain sequences. It can produce the motor behavior representing its beliefs. It can also has some other tasks, and we'll look into it afterwards. So The question from Mario Roman Cabezas to everyone, so everybody can answer, is how can they map that to parts of the brain? The answer is they assume what different parts of the brain are doing according to what neuroscience has informed, and where there isn't enough information, they interpolate or extrapolate or pull out of their innards what it should be doing. So for example, the pattern recognition problem is resolved at the level of the visual stream. So he has models of V1, V2, V4, and IT. So that at every single level, the pixels that are coming in via visual system are transformed into higher level percepts. We're going to definitely get into that, particularly in our final lecture, lecture 5. We're going to talk a lot about pattern recognition. But so Annabelle Horst asked, this is not a learning model. This is not a fully plastic model in which the brain is led to do what it wants, but this is indeed a learning model. As you see, for example, he can reproduce sequences and he has learned a certain sequence. And you can, in fact, teach this model, only that it's a little tricky. It is not, the model doesn't decide when he wants to learn, but the experimenters decide when the model is learning. So it does have sort of like button presses, like do paradigm 1, do paradigm 2, which is not different than telling a person what task is it that the brain is doing. So for example, if I ask, you know, like can you draw a dog? You go ahead and draw a dog. So in the model, you do it a little bit in the same way. You have a input that represents the query for the brain to do something. And in order for the model to know what a dog was, he has to be trained on what a dog is. And so there are certain protocols that have been used for that. So it is still a learning model in the sense of deep learning almost, in which you have types of backpropagation and spiking networks, but it is not a model with intrinsic plasticity that is learning at every single iteration, at every single moment in the computational brain. I hope that that was clear. So the answer is, this is a learning model, but is not a fully plastic model. And so this brings me to the last slide of this flyby. So the departure point, the departure point of the two is similar. It's the neurons. So we think brains work with neurons, but neurocomp focuses on function, what can it do, and computational neuroscience focuses on biophysics. So how does the machinery work? The assumptions about what it's included in a model, and therefore its complexity, commonly varies. Computational neuroscience says, like, you didn't put that ion channel in, and the neural computation says, like, that ion channel doesn't matter. The explanatory goals differ. For the moment, they've been diverging, but a holistic understanding of the brain surely must bring the two together. The word consilience that titles this slide has been brought into the populace by Edward O. Wilson, and he was talking about the bringing together of multiple threads of knowledge. The idea that once you produce knowledge in divergent paths, they eventually, if they're talking about the same thing, the brain, they have to converge. So we will eventually find out which channels matter, or what is the role that the channels, ion channels, have to play on certain behavioral cognitive functions. So this is the end of the computational neuroscience versus neural computation part. Would you like to ask some questions? This is a perfect moment. I'll give you 20 seconds. Now it's 11 o'clock, and you can ask questions as I explain what's going to happen next. We're going back to our the lecture itself. We start with computational neuroscience. We start with what in the neurons produce action potentials. And we're going to then, I'm going to be talking a little bit about neurons, biological neurons. I don't have to talk much. 90% of you are confident that you know what a neuron is, and probably even synapse. I didn't create a question specific for synapse. Synapses are the points of contact between neurons where the activity is transmitted. I don't say information, notice. And so I'm going to move on to neurons, and I'm going to move on to computational neuroscience view of neurons. And after that, we're going to talk about the project itself. So my plan right now is to walk you over very high level what is relevant in computational neuroscience, and then go back to the project. There were no questions while I was delaying this moment, so I just carry on. We're having another pause just before I start talking about the project, okay? So in, I think, another 15 minutes, we can have a pause. Abstraction. So this is a diagram of a neuron, how we envision it. There are other kinds of neurons. This I took from the Encyclopedia of Neuroscience, and this is the canonical neuron, the mindset that we have. And we think about it as a single element, like floating in space, where you have it's receiving information from its dendrites above, integrating this information in the nucleus, and sending this information down the axon with a very fast signal through the myelinated axon to the postsynaptic dendrites of the postsynaptic cells, postsynaptic neurons. So this is the canonical view of how a neuron is connected to his other neurons. Notice that I said, so I used some neural computation words here. I said when inputs come into the dendrites, they're integrating something, but what they're integrating is, in different views, different. Like you can be simply adding membrane potential, or you can be integrating information. Here we're talking about computational neuroscience, yet I've used the word integration. The reason for me to start with this diagram is because I'm not going to go into all of the details of the neurons, but that ever since Ramon y Cajal, like the first guy to have actually drawn single neurons and think about neurons and as individual units in the system, we started making a conception of neurons as isolated cells that are kind of like floating in space. They're fairly sparse in the brain. In this drawing that you see on the right side with a Golgi stain, which is big backstory. I'm not going to go into that, but it's a Golgi stain. You see that the neurons they appear fairly sparse in the brain, and so we started thinking about them as individuals. So Cajal helped us shape an understanding of the brain that focuses on the single neuron as a particular individual. Modern neuroscience has brought back some understanding that these neurons are actually very thick. They derive evolutionarily from skin tissue, and so they're very dense in the brain. Here I use this green fluorescent protein slice that has some I think these are Golgi cells. I'm not super sure, but these yellow cells. Notice that there are here some black spots. I hope you can see my mouse. There's some black spots in the tissue. It means that the cells are completely densely packed together. So we also have to think about neurons not only as individuals, but also as being a part of tissue, flesh. It's like fatty tissue. So they're not acting isolatedly, but in a group. When two neurons are close, they most likely receive very similar dendrit axonal arborizations from the neurons that are informing them. So the closest two neurons are, the more likely that they're going to be doing similar things. And this is something that has been seen across many, many brain areas, well, not all, but many, many brain areas, and restitutes a view that the tissue is important. That's why we look into local field potentials as well. Lower dimensional systems. So you see how packed it is with fibers, right? This is ready to give us neurons pictures. So this is the brain, but which neurons have different colors depending on their protein makeup. And you see these fibers and just a few somata, but in fact you have to imagine that this is completely packed with it. So it has a certain probabilistic existence. It's not completely isolated as Harmonica Hall made us believe. So let's think about them as populations of neurons. The more we think about neurons, the more we have to think about what they do in coalition. And how they convince each other to work in coalition is via spikes and synapses. So you may have heard that by applying a current to a neuron via an electrode, here we have a current generator. So you have a membrane current that creates voltage deflections of the membrane potential of the neuron. And so here at different membrane currents, first bump has a certain amplitude, a square pulse that doesn't quite produce an action potential. You give a second bump that is a little bit larger, produces a slightly larger action potential. Notice that it doesn't simply copy the square, but it has this little curvy low pass. And then with a little bit larger input current, you actually see this all or none event, the spike. So, might be led to think, oh, well, so, it's either or. I mean, it either spikes or doesn't spike. That's an oversimplification, and computational neuroscience is going to be looking into what causes a spike. But this is the ancient conception of spike production. You either have subthreshold input or suprathreshold input. It's important to realize that the neuron has a state, and what is suprathreshold or subthreshold is going to depend on what state the neuron is in. This is what Project 1 is also going to be about. Also lecture 2, we're going to be looking into the mechanisms of spike production and enriching the view that what the neurons are doing is simply input-output functions via threshold, simply infinitely short pulses. It's more complex than that. It's more interesting than that. It's richer than that. But nevertheless, this is the image that we all started from. So we take the Hodgkin-Huxley model as an explanation for how spikes come about, and that's what Project 1 is going to be about. So this is the anatomy of a spike. This is, again, what Project 1 computational neuroscience is going to be about. We're going to be discussing in a moment. You're going to be explaining where a spike comes from on the basis of ion channel fluxes. This is the equation we're going to be breaking into pieces and looking into different components so that we can reassemble this model from scratch. So it might be daunting as you're staring at it now and saying, whoa, what? And I'm not even showing you all of the equations that play a role here, but the model and the project we have subsequently are going to break into pieces so that you can focus in each of these variables independently and know for sure in your heart that you understand this equation and why it is so revelatory, why so powerfully explanatory about the action potential. I might not have mentioned that the action potential was discovered in the squid, with tentacles, in the axon of the squid. That's the experimental problem that Hodgkin and Huxley were studying as a model for even mammalian cells. This has been used, which is amusing to think about. at the level of generalization. So it's important to understand the Hodgkin-Huxley neuron because it underlies basically the human brain project. All of the models are Hodgkin-Huxley type neurons. They have more complexity and certain differences, and important differences. They're also multi-compartmental, but their core is this. So if you understand their core, you will be equipped to understand all the way until that simulation of the brain that we just looked into, or at least so I hope. But you don't need to necessarily look into all of this multi-dimensional complexity with multiple parameters. The types of spikes that have been observed in real life are many. So here you have many kinds of neurons. Here, these are the activities or spike patterns of different kinds of neurons, and they can be explained via simpler models than the Hodgkin-Huxley. We're going to be dealing with simplified models of action potential on our second lecture, where we're also going to look into very simple models of spiking. So here, just to give you a sense of what this figure is for, we're going to look again into it with loads of detail in lecture 2, but just notice that you have two traces. The top trace, which is the membrane potential trace, the spikes that the neuron is producing, and the bottom trace, which is the input current, what is coming in through an electrode. Could also be representing synaptic input. So you have different kinds of neurons. Focus, for example, simply in A and B. If there can be neurons that, upon the exact same current presentation, one neuron is going to start spiking robustly, pum, 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 and the other one is only going to produce one single spike and be happy with it. So this is the kind of difference that exists in spiking between different neurons. Many others exist. We're going to discuss them in the detail, as I mentioned. And underlying all of these spiking patterns from Hodgkin-Huxley and other models are the neurodynamics of these models. So in lecture 2 also, we're going to be talking about the analysis of membrane potential variables and gating variables, as they will explain the different spiking patterns. So first you have to go through project 1 to understand a little bit about what the basic simulation model of a single neuron is. Then we're going to simplify and try to reproduce a larger spectrum of different spiking types. I'm going to give you a sense of this. Like, this is a fairly deep field, neurodynamics. We treat it in dynamical systems in the masters for nanobiology. We look into attractors and things like that. And here I want to give you a flavor for that will help you explain for different kinds of neurons, what are the conditions of creating spiking, and what are the conundra, what are the difficulties with that procedure. And this was the last slide of that moment. So now it's 11:15 as planned, and we can go into the project. I was going to suggest we have a little bit of a pause, and then then we return to Google Colab for writing your first do-it-yourself action potential for a spike. So see, 11:15. So if we start again at 11:30, I guess that would make sense, and then we can go until 12:30, 12:45 working together, right? So like after 11:30, we can have another hour and 15 minutes of joint work. Just before we go there, let me just say, you're probably not going to have the chance to finish the project here now, but the project is it exists by itself, so you can go through it by yourself, and we can then ask and answer questions in the quiz next lecture. Okay, so 15 minutes now, I hope. So 11:15, physiological pauses and electrophysiological pauses if you need. So we reconvene at precisely 11:30. All right. So welcome everybody, and to your own do-it-yourself Spike project. Where to begin? So there's a couple of things I need to say. So you probably, if you ever stumbled upon Python before, you heard about Anaconda or Jupyter Notebooks, for example. So Anaconda is a Python distribution, and you have to install it so that you have access to all the marvelous Python packages, and that you can use, all of the Python environment. In order to program Python, you have to have a Python installed, and Anaconda is a Python distribution. And it's painful sometimes to install. So we decided to instead to use Google's lovely solution that they are actually using quite a bit also for artificial intelligence. It's called Google Collaboratory. And as you can see in the link, it says collab.research.google.com. So Collab is basically shared Jupyter notebooks of Python. You'd have sound but lower quality. Okay, so potentially, let's see. Yeah, so the right microphones have been selected. Okay, so maybe that's a local problem then. Okay, so maybe come back in a moment. It's still recorded, so someone was complaining about connectivity issues. Okay, so Jupyter notebooks then are these lovely abstractions of how to create text and code at the same environment, so that you can follow a stream of thought while you have parts of this notebook with explanations, and you have other parts of this notebook that are code. And you can run the code, and the code will then execute in the background in this distribution. Someone is listening to music with their open mic, so I would appreciate if you if you close the mic as you're listening to music. It's okay listening to music. Nice background. Anyways, and that's this. It allows for a lot of structure. So if you look here on the left, it has a table of contents, and this is hierarchical. So the tab indicates which section of the notebook we are going to be looking into. It allows us to, for example, collapse certain blocks of text. So here, project 1, and you click on the little triangle, and you get the explanation about what the project is like. Ta-da, ta-da. You will be able to copy it. So I'm first explaining what this notebook abstraction looks like. So you have then this text part, and the text is written in Markdown. If you double click a part that has text, you will notice that it looks a little bit like LaTeX or LaTeX for some of you that might have stumbled upon it. Is basically an encoding for text that creates not hypertext, but creates formatted text. So here these so hash signs, they indicate the hierarchical structure. So if you have one dash sign, this is going to be at the first level of the hierarchy, and if you have 3, it will be at the second level hierarchy. So I'm just explaining how text works here. So this text you're reading, underlying, is coded in what's called Markdown here, Markdown. And if you're curious, you can look into it later. It's a lovely way to write formatted text also with math, as you will see here. Then all of the projects will go through their own explanation. So they're self-explanatory. The idea is that you can go through them by yourself once you understand what Collab works like, and really structuredly learn about this topic, which in this case is the Hodgkin-Huxley model. So here's some key terms with their explanation. So if there's something that you need to brush up your knowledge first, you will also find out here in the prerequisites. Prerequisites also have some contributed videos from some of the teaching assistants and people that have helped me putting together this project. I want to explain this line. So this is the first time that we are actually encountering some code in this Jupyter notebook. And so you see here, there's this text part, some bullets. And here at the bottom, you have some Python instructions. The first line is actually starts with an exclamation mark, and it is telling Google Colab to execute a terminal command pip to install a Python package called Brian2, which is effectively the simulator that we will be using in this project. There's a lot of stuff you don't need to know here, and you don't have to worry, really. You can learn as you go, because most of the code does not require this, and you have also example code that is already working. The idea is that you will read through this code and learn as you're reading. So these are the packages we are importing. We explain a little bit about which packages we're importing. You can then collapse this again, and then you don't need to look into it. But what you do need to do, first and foremost, is to press, for example, what you have to press is Shift-Enter to execute that piece of code. You could also have simply clicked on the play button here, and that would then execute that code. And in the background, and you can see here, it's collecting Brian, and it's basically these things have already been installed, so these packages are already there. So you get some feedback, as you would get, for example, from Anaconda as well in the Jupyter notebook, and so forth and so on. So that means that now you have your necessary packages loaded. That's why this cell here is called initialization code. For every code that you want to run, you have to either press Shift-Enter on this code block, or you have to press play. And the whole code is sequential. So the sequence of your pressing plays is actually very important. So if you go to the last code bit and you press play, it won't work because it depends on the one that is just before. So this is something important to remember, that it's executed sequentially, like in MATLAB. So here in the background, there is a workspace, for those of you familiar, that include all of the variables that have been initialized and the packages that have been initialized. So if these things seem daunting, the first thing you can do is you can watch this video before you even go here. You can watch the video on Google Colaboratory, and then you understand a little bit more about how it works. It's up to you how you want to go about this, okay? So I recommend read through the instructions of the project. So, so the key terms. Do read the learning objectives. This is what I expect that you will have learned after you have gone through this project. So you have, you're going to learn about passive ion channels and active ion channels, certain things that I have not talked about in the introduction. Okay, so we have here a question. So if you edit the very first cell, you can see that the video actually does not have a URL connected to it. It's just an empty link. Is that true? So did you guys, can you guys go to the link I shared? Which one is, you mean the edit the first cell, the video on the Google Colaboratory is not there? Okay, let's see the link. So, oh, that's why. Now, it was supposed to be here. Thank you, Lars. So now I'm going to Google it after we finish this. I'm going to Google it and put it there. Sorry for the lapse. By the way, it's the first time we're deploying this version, so you might stumble upon certain problems. That's why we're going to be there for you. Okay, so there are 4 people here that can give us support. Some of you are actually taking the lecture, like Roger Small, but I wanted to introduce you also to Natya Shamuja. She's not an nanobiology student, but she is working with me on these projects, and she's quickly becoming a Python expert. She actually might even say hello. Hello, Natya.
Hi everyone.
And there's also Daphne Cornelissen. She's also a student of mine. She's working on her capstone project by the Erasmus University College in spiking neural networks appropriately. And she's also going to give us some support. So Daphne, if you're listening, maybe she's not listening.
I'm listening. Hello.
Oh, very good. So, and so we're going to go into Discord, and we're all going to give you support if the project has issues, or if you have issues with the project, or any other general issues that might appear. Okay, so the Google Collaboratory video is going to be put in a blink.
Can I say one thing? I noticed that
Yes,
you can also just click on Save, Copy, and Drive, because the instructions say that you can click File and then open in Playground, but you can also just directly do Save, Copy, and Drive. I had it as a button, but
okay, great, exactly. So you see here that you can make a copy, a local copy, by copying it in your own drive, and you can also open in Playground mode, which is, I think it doesn't even require you a login, but I'm not sure.
No, I don't think so.
Yeah, so that's nice, because then we are GDPR safe here. If you're not willing to share anything with Google, you just go into open in Playground mode. If you really want to work on your project like enduringly, then best to save a copy in your own drive and move on from there. Thanks, Daphne. Let me now go through the structure of the project itself quickly. So we have here these key terms, the learning objectives, and this project is broken in 3 pieces. We're going to be talking first about what happens at the level of the passive membrane when we're exchanging ions and changing the membrane potential. Then we're going to look into active ion channels. You might have heard the word before. I haven't checked, but there is an explanation of what ion channels are here as well. And if they're missing, come to us in our Discord support forum. And then you effectively are going to put together a Hodgkin-Huxley model, the entire thing, as a last thing of this project. Then there is a, actually, let me open one of my own Google projects to show you. So after you go through the entire project 1, you will have a model. You will have a model of a spiking neuron, a Hodgkin-Huxley model. And then you will go to this extra project or extra part of the project where you can use the model to do experiments. And in this model here, we will be reproducing the picture from Ijikevich's book in 2007, the dynamics of spiking, or the dynamics of what is it? Geometry of birth thing. Dynamical systems in neuroscience is the name of the book. And we're going to be reproducing this figure because it's the anatomy of actually what creates a spike. And I believe that it's absolutely fundamental for everybody here to, in the end, be able to tell what in the cell we believe, like model, create a spike. And so you will be reproducing this by finding the parameters that actually produce this picture. So then you have here a little bit of code. Much of this code is given to you, so you don't have to worry about recoding it from scratch. So what I want is for you to read it. You read it and you understand. So that's, and there's some questions along the way for you to ascertain that what you believe is correct. There are also some answers along the way that you can open yourself. How is this project graded? Not at all. The project is so actually we didn't talk a lot about testing, right? So just as soon as I finish talking about the project, we're going to talk about the project presentations just for 3 minutes, and then we can have more questions in Discord about that, okay? But the project is not graded. I want you to do it because you want to learn. This is an elective course, and it actually brings people of different backgrounds. So I don't want to impose on everybody the same criteria. But I think that the level of information that I provide is such that you can look at it from different scales. So you can look at it from, very detailedly, like doing the code by yourself and creating powerful experiments, or even creating further functions that elaborate on it to answer some of the questions, or you can simply mostly read it and understand it, because this is Python. I'm not forcing Python on anyone. I want you to learn about Python by reading it, by seeing the powers that it has. I mean, like I am also a MATLAB, I also like MATLAB. I think those are both excellent languages, but Python in this scenario, you have this amazing advantage of being online, and you don't need a server to compute these models, and this is a substantial advantage. So not graded. It's for your own benefit. We will be discussing the Hodgkin and Huxley in a quiz in the first thing, then the next lecture. And the nice thing about it is that if everybody goes through it, then the quiz is going to be super short. And I think that within a week, you will have the time to do it. You're also going to get some support at the Brightspace forum. Okay, so like there will be a Brightspace forum in the primary neural networks Brightspace where I will be answering specific questions about the project. Okay. And the Discord page will be open. So I will be addressing the Discord page as well throughout the course. So the Discord is going to be open. I might not be there all the time on the fly, but if you leave a question, we will see it. You will join the Discord when I give you the link. Haha. Will you upload the course schedule and exact grading scheme to Brightspace? I think that the Brightspace has it already, if I'm not mistaken, unless I'm much mistaken, because here in content, in course information, assessment, you don't see both of them. One of them is hidden. You only see assessment specification. And this is it. You'll be evaluated via presentation and a written report, 50-50. The projects will be groups of 1 or 2, but the projects are individual, and you get one joint grade for the presentation and an individual grade for your report. Will you upload? So I guess that this covers it, so I'm hoping. And if there are more questions, please just join the Discord. Okay. And so this is, the project is self-standing. You can now copy it to your own and start there. I'm going to load up that link that is missing on the video. Maybe there are also other videos missing. And please ask a lot of questions in the Discord. I'm going to give you now the link. So because I had to restart the computer, it's downloading updates apparently. So I'm going to then send you the link to the to Primary Neural Networks Discord page. You can also open it in the web. You don't need a login. You can also do it by yourself in the background. So here it is, our primary neural networks. I have to be able to share it as a link, invite, and then this is our link, and this is it in Zoom. Ta-da! Welcome. Welcome to our Discord forum, everybody. So if you have troubles, let me know. Now, since it's already 10 to 12, that means we're not going to be having a lot of time to work together. I would suggest we work for another hour together until 12:45, and then you continue working. And I will be there in Discord and checking your questions and answering here. Let me just walk you through the Discord page a bit. Okay, so why is the, okay, so here. Don't know if you've been to Discord before. These are multiple chat rooms, basically. So you have here text chat rooms, which are called general course channels. One of the announcements, for example, links that I want to share, general announcements about the project themselves. We have some general questions, you know, some maybe related or unrelated. I love the brain. Well, how do I open up a skull? Something like that. And then you have the raise your hand channel. In the raise your hand channel, basically what you do is you go to it and you say, I have a question. And then I know that I have a question and I can talk to you directly in person. Or one of the TAs can come and talk to you in person. When the TA sees that, when a TA sees a question, the TA can open a chat, and then he would come here, mark it as with a thumbs up, saying, "I have seen this, I've taken this up," so that the other TA can, continue to make himself available. So basically, it's a matching system, right? So one person raises his hand, and one TA comes and says, "Hello." It could be me also. And then here we have some student spaces. You can self-assign. So if you just click in here in the study group 1, let me see here, study group 1, notice, okay, I got it. So now I'm in study group 1, okay? If anyone else clicks on study group 1, we will be there together. And when I speak, they will hear. So this opens a devoted channel for a certain group. So you can then self-invite. Yeah, so this is the chat. This is study group 1 text channel. And right now my microphone is off there, obviously for obvious reasons, but as soon as we close Zoom, I'll be available there, and we can also create these one-to-ones. So I see you coming in and out of the channel, and so this is where we are together. Couple up if you want to work together in these projects. It's great. You can also ask each other questions. You don't need to ask us. If you already have people that you want to work together, you're free to do so. Use these spaces, right? So that the idea is that this helps us self-organize and match with our desired peers. Questions? Questions about Discord, about the project, about the course. I even have some unreads, new unreads. Let's see. I also have a secret channel. And then you can, I guess that right now, is someone asking questions through? So secret TA. Oh, voice connected study group 1. I see. Okay, so everybody's in study group 1. Okay, great. So this is working well because there are no further questions. I was going to then wrap this up and move into Discord with everybody together. And so welcome to Primary Neural Networks. I hope you enjoy the course. It's been redesigned from scratch to address the Corona challenges, and I hope you enjoy it. It's been a confusing experience so far for me. I hope you were not too bothered by the delays in the beginning. And we go aan de slag, as they say. So I'm now closing the Zoom channel, unless anyone else has a pressing, desperate question. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. See you soon. See you in Discord.